Hi, this is Vedika. I'm a research fellow at Pine Biotech and I'm also studying for a master's degree at Amity University. I'm here to talk about my project where I studied the genetic characteristics and the role of tumor microenvironment in chronic lymphocytic leukemia, which is also called as CLL, through transcriptomic analysis. Chronic lymphocytic leukemia, to begin with, is a chronic lymph-proliferative disorder, which is characterized by an increased production of monoclonal B cells, which are morphologically alike and look more on the mature side, but they are immunologically dysfunctional. To perform their humoral immunity roles by producing antibodies, and hence they are immunologically dysfunctional, and they also have a smudgy appearance on the peripheral smear. So mostly genetic factors rather than environmental factors are responsible for the occurrence of CLL, but few environmental factors such as occupational exposure to certain chemicals and radiation, and also the use of tobacco can lead to the occurrence of CLL. Coming to the symptoms, mostly patients with CLL are symptomatic at initial presentation, and a routine CBC just casually reveals abnormal lymphocytosis, which occurs when there is a population of greater than 5,000 cells per millimeter cube of the blood sample. Approximately 5 to 10% of the patients with CLL are symptomatic with B symptoms such as fever, unintentional weight loss, fatigue, etc. So the pathogenesis of CLL can be mainly divided into two steps. Step 1, where there occurs abnormal clonal division of neoplastic B cells within the lymph nodes. And secondly, the increased B cell lymphocyte production, which are morphologically clonal to each other. They are released into the peripheral blood, leading to the diagnosis on CBC. B cells are very important components of the immune system as they contain the B cell antigen receptor, also known as BCR, on which lots of molecular therapies and precision medicine based therapies are being designed to cure and treat CLL. It's responsible for the induction of antigen and autonomous signaling within the cells. Also, as I talked about the occurrence of uncontrolled clonal division of B cells, it can occur due to various primary factors such as antigenic stimulation, genetic mutations, and previously existing genetic abnormalities. But the MBL can progress to chronic lymphocytic leukemia by the further insult to the B cell clones due to genetic abnormalities or occurrence of infections or changes within the bone marrow microenvironment. This tumor microenvironment is receiving lots of attention when it comes to its role of within cell signaling and the occurrence of normal immune responses. That's why the tumor microenvironment is also being looked at as one of the causes, and its defects can also be used to develop therapies against CLL. The tumor microenvironment represents a highly complex cellular component, including non-hemopoietic stem cells, extracellular matrix, lymphocytes, and myeloid cell subpopulations. These elements confer therapy resistance by creating an immunosuppressive environment. That's why the question or the goal around which the project is designed is to understand the molecular mechanisms behind the interdependencies of intra-tumor heterogeneity and tumor microenvironment in CLL. So for this project, the dataset labeled GSE 161711 was obtained from the GEO website. 152 samples were obtained from 37 patients, and this number of samples exceeds the number of patients because each patient gave 3 types of samples. They can be broadly categorized into 2 categories, namely peripheral blood, and the other one was from the lymph node. The peripheral blood sample only contained CD19 positive cells that were filtered and extracted, and the lymph node gave 2 kinds of samples, namely CD19 positive cells that were filtered and isolated, and that group was called LN, and the whole lymph node biopsies that was LN mix, where a mixture of other kinds of cells such as T cells also present in the lymph node and its matrix was also obtained, which was sequenced, and the reads with a quality score of less than 1 were removed. The second step was to perform a PCA and statistical analysis so the genes that are significant and significantly expressed could be analyzed. PCA is a dimensionality reducing tool, which helps us look at multiple components of data simultaneously. And also helps us know the variations within expressed transcripts at a baseline level. So the PCA was performed using the T-bioinfo server pipeline, and it is a very interactive pipeline. Then using Excel, we calculated the p-value, the p-adjusted value, just to make sure that no false positives were obtained when it came to the expression of genes, and log2 fold change, which calculated the differences in the quantitative differences in the level of gene expression for all the samples. The third and the most interesting part was to perform a differential gene expression analysis, which actually helps us understand the quantification and the significance of raw reads, how they were expressed, to statistically significant results, and also helps us correlate to their molecular pathways and the functions of the genes. So about 34,841 genes were selected for further analysis. Within the lymph node subgroup, a log2 fold change filter of greater than 4 and negative 4 was applied. Within the CD19 positive subgroup, a log2 fold change cutoff was applied as less than -2 or greater than 2. The David tool was used for us to be able to understand and correlate the gene IDs with the ensemble gene IDs, which is a more specific way of looking at genes, and also being able to correlate them with their names and functions. PantherDB was used to perform gene ontology analysis, which helped us look at the pathways associated with the genes, their functions, and also the molecular functions and biological functions of the same. So the results were as such. Firstly, we got a PCA plot for both the subgroups. That was the CD19 positive subgroup, where we had looked at peripheral blood versus lymph node samples. As you can see that the PC1 component was 18.68% and the PC2 component was 6.68%. The PC values for the lymph node group are shown with the help of blue triangles, and we can see that they are mostly concentrated in the second quadrant above the x-axis, and the peripheral blood ones, they are kind of more densely present within the third quadrant and are denoted by red circles. So this helps us understand that there are differences in the transcripts that are expressed within both the subgroups. Then a PCA scatter plot was obtained within the lymph node group for CD19 positive cells and the lymph node samples that had all kinds of cells within the mixed group. The PC components remain the same, and we can see that the lymph node CD19 positive cells that were obtained from the lymph node are more concentrated in the second and third quadrant on the left side of the y-axis, whereas the lymph node mixed cells are more concentrated in the first and fourth quadrant away from the y-axis. So this also shows that the transcripts that were expressed in both the subgroups and the genes that were significantly expressed, which can help us correlate to molecular mechanisms like transcription, gene expression, gene repair, etc., are different in both the subgroups. Then moving on to the statistical analysis, for the lymph node subgroup, after applying the appropriate filters, about 1,103 genes were differentially expressed. They were found to be of such number. 1,101 genes were upregulated and only 2 genes were downregulated. Within the CD19 subgroup, after we applied the appropriate filters and filtered out the most significantly expressed genes, about 387 genes were massively differentially expressed. 353 upregulated genes and 34 downregulated genes were found. And here's a snapshot of the David tool, how it looks like and how we are able to correlate their ensemble IDs to their gene IDs and the names of the genes. So the differential gene expression, when we were able to look at their p-values and p-adjusted values in the log2 fold change, which all helps us understand how quantitatively how much of the gene was expressed how high or low the gene expression was, and how significantly it was expressed. With the help of the Volcanos 2R, we drew a volcano plot, and as we can see that the downregulated genes are expressed by blue dots, and the upregulated genes are expressed with the help of red dots. We put them on a graph where the y-axis shows the negative log 10 value of the p-adjusted or the p-value, and the x-axis talks about the log fold change. For the CD19 subgroup, we see that certain genes were the most significantly expressed and most upregulated, such as the AMI1A gene, which is the alpha amylase pseudogene involved in the cellular oligosaccharide metabolic processes, and the TMEM53 gene, which is the transmembrane protein 53, which is responsible for transmembrane translocation of various biomolecules, and the MRPL20 gene, which is the mitochondrial ribosomal gene, which regulates the translocation of mitochondria. The PIK3CD gene, which is the phosphatidyl inositol 4,5-bisphosphonate 3 kinase, and it is the catalytic subunit of this enzyme, which is involved in B cell proliferation, development, and migration of the B cell receptor signaling. And it also helps the T cell receptors perform their signaling functions better, and also is responsible for a very interesting function, which is the antigen dependent memory of B cells. Also, some small nuclear associated RNAs such as LSM10 and non-coding RNAs were also found that are involved in tumorigenesis with the regulation of gene expression and their role in differentiation of cells by regulation of their receptors. The most downregulated genes involve TNRP1 and ACOT7, which is the acyl coenzyme A thioesterase 7. It is a protein coding gene and encodes a member of the acyl coenzyme family, which is very essential in the fatty acid metabolism. When we talk about the pathway analysis plot of the CD19 gene, we see that the pathway is most significantly overexpressed or most significantly associated with the occurrence of CLL for the samples of the CD19 plus subgroup are the North signaling pathway, and which is associated with the interactions of tumor cells with the microenvironment, angiogenesis pathway, the Alzheimer disease penicillin pathway, and the HF receptor signaling pathway, and the P53 pathway. When we talk about the molecular functions plot, we see that several molecular functions are associated with the differences occurring within the lymph node and peripheral blood subgroup, which is the ATP dependent activity, such as the chromodomain helicase DNA binding protein 5 that binds to the DNA via histone protein, and it regulates gene transcription. ATP hydrolysis microtubule binding, and catalytic activities such as phospholipid binding, signal receptor binding by phospholipase A2 family, and other members of the cyclin-dependent protein serine threonine activity. Now, moving on to the lymph node subgroup, we see that in the lymph node subgroup, a similar volcano plot was drawn where the upregulated cells were denoted with the red circles, and the blue circles were responsible for the denoting of the downregulated genes. We see that several top upregulated genes, such as MTHFR, which is the methyl tetrahydrofolate reductase, was overexpressed to a log fold change of 6.68 times and is involved in modified increased risk of CLL by increasing homocysteine levels. The EF hand calcium binding domain 7 is denoted by the gene EFCAB7, which is responsible for positive regulation of RNA polymerase II dependent transcription. The pink1 gene, which is the P10-induced kinase gene, encodes for P10 and protects cells from stress-induced mitochondrial dysfunction. Also, 2 to 3 genes were downregulated and were observed in this group. Coming to the pathway analysis plot for this group, we see that the pathways that are responsible for differences within the lymph node and the lymph node mixed cell population, they are because of biological processes such as signaling, cell-to-cell adhesion, controlled transformation of phases of the cell cycle, checkpoint regulation, T-cell regulation, growth of cells in activated and proliferating states. Interestingly, lots of immune functions such as activation of the CD4 plus T cells and cell-to-cell signaling in antigen-presenting cells and lymphocytes was also observed. The pathways that were most commonly associated and most enriched within this subgroup also included the Notch pathway. The Notch pathway is one of the most commonly observed pathways that acts by regulating the tumor microenvironment and also determination of the cell fate. Other pathways such as WNT signaling pathway, the JAK-STAT pathway, and the P53 pathway was also observed, which is shown to influence tumorigenesis and is seemed to be activated by microenvironment molecules such as CD40 and epitopes like that. So the principal component analysis is a dimensionality reduction method used for large data sets. And on the whole, to summarize the project, significant differences were observed in the tissue types such as lymph node and peripheral blood within the CD19 expressing samples, indicating microenvironment differences. Within the lymph node tissue biopsy, samples of most points and patients within the CD19 plus and mixed lymph node cells were variable, indicating differences in somatic mutations and the interplay of immune regulation. These observations of statistical analysis identifies 1100 genes within the lymph node subgroup and about 3700 genes in the CD19 subgroup that were significantly downregulated or upregulated. It's because of the recent advancements in NGS and genome-wide sequencing that have led to the understanding of constantly mutated genes that play a role in DNA damage repair. For example, TP53, chromatin modifications such as HIST1, H1E, and pathways such as the Notch1 signaling pathway, the WNT pathway, and various other inflammatory responses occurring in the B cell. The functional role of myriads of putative driving mutations have been confirmed as gene silencing that results in decreased cell viability and an uncontrolled clonal expansion of immature B cells. To conclude with, the goal of this project was to understand the upregulated and downregulated gene molecules, their associated pathways, gene products, and cellular functions, so that new therapeutic targets and clinical markers to predict the diagnosis of CLL patients could be understood. We noted that CLL is an indolent malignancy characterized by an increased accumulation of CD19 positive and CD5 positive expressing cells. The tumor microenvironment certainly plays a very important role in the prognosis of CLL. Gene signatures indicating the crosstalk between genetic characteristics of CLL along with those of the immune microenvironment have been characterized by this transcriptomic analysis. They play a role in several cellular pathways, and these mutations can lead to a wide range of cellular consequences, such as aberrant DNA repair, altered BCR signaling, and an enhanced inflammatory state in general. The critical molecular pathways, such as Notch1, which was the most expressed pathway in both the subgroups and drives chemokine signaling in the tumor microenvironment, it confers disease refractoriness and cell proliferation, along with other pathways such as the WNT pathway. And these findings provide a fundamental information for understanding the genes and pathways involved in pathogenesis of the immune microenvironment that drives CLL and their role as therapeutic targets and biomarkers should be studied in vitro as well as in silico. Thank you.